Andrea, thanks for joining. Thanks everyone who is here today. Let's see. We're now competing with another meeting, a professor's meeting here, the faculty assembly. So that's the reason why many people are going to be joining late, but I hope more people is coming.
That's no problem. Thank you so much for inviting me.
Yeah, it's a pleasure to have you here, Andrea. Well, especially have someone from our country. Yes.
Hi, Dr. Gladys.
Hi, Andrea. Good to see you, and thank you very much for accepting our invitation to be here with us.
Thank you.
How
Thank you
is
so
the
much.
weather over there?
Well, it snowed once on Saturday, and my mail got delayed for 5 days.
Wow.
Things like that are happening over here, but it's okay compared to Colorado. My car battery didn't die suddenly, so it's all wins.
Okay, good.
How are you? How's the weather over there?
Well, it's very nice. It's kind of cool and breezy, so I like it very much. It's good to take a walk in the morning, and so yesterday was beautiful, actually.
Oh, that's awesome. I have not been yet to Brownsville. Hope to visit
Yes,
one of these days.
yes, you have to come and see the valley. It is
Yeah.
really beautiful.
Yeah.
So, Anne, I think we should begin.
Yeah, no, definitely. Yeah, in order to start
Yes,
some time and to do a good use of the time, let's start. We are recording. Is this okay for you, Andrea, to record your?
That's okay. Yes.
Perfect. Okay, that's nice. We are going to be recording. We have today Dr. Andrea Mendez. She's an MD, PhD from Maracaibo, Venezuela, where many of us are also. And she's a neurology resident at Duke. She got a PhD in cognitive neuroscience from Colorado State, where she do research on white matter decline and plasticity in Asian populations using MRI. At Duke, Dr. Mendez is dedicating her research to address neurocognitive disparities in Asian and promoting health equity in the Latino population, something very close to things we are doing here. So I would like to say a big welcome to Dr. Andrea Mendez, and let's go with you with your presentation.
Wonderful. Thank you
Okay.
so much again for inviting me. I am going to share my slides here in just a second.
Yeah.
As I prepare my slides, if everyone could type in the chat, if you have a second, what is your current position, whether you're a postdoc, faculty, resident, med student, that would be great so that I can have a better sense of and an idea of everyone's backgrounds.
this is a very friendly group.
Wonderful.
Yes, it's and very insightful, as you would see, Andrea, and very multidisciplinary,
Right.
from really basic scientists to clinical scientists, clinicians, and of course our very and ethics, and also our very supportive uh, research associates, which of course we rely on to do our work. So,
Right. Fantastic. Wonderful. Thanks for everyone for writing in the chat. I'm going to prepare my pointer here. There we go. So I have titled my talk, Advanced MRI, Lifestyle and Brain Health, and Understanding White Matter Plasticity in Cognitive Aging. But I'm going to spend a good amount of my talk talking a little bit about MRI and background related to white matter so that we can better understand in the context of aging as well as Alzheimer's disease and related dementias. And as Dr. Ali mentioned, I am currently a neurology resident at Duke University. So I'm a first-year neurology resident, meaning that I started residency 6 months ago, and I'm halfway through my intern year. And I'm also a research scholar for the NIH R38 program. And that specifically is a program that stimulates access to research and residency. And because my goal is to become a physician scientist in the field of behavioral neurology. So to jump into the talk, this is a little bit of what we will talk about today. So as I mentioned briefly, we will talk about Alzheimer's disease in the context of the brain's white matter. Hold on, let me move this. Okay. We will understand a little bit of the white matter structure, the white matter of the brain. We will mention some MRI techniques that we use to study the white matter, some of the traditional and some of the new techniques, and talk a little bit about how the white matter relates to lifestyle, particularly in the context of physical activity or exercise, and show some of the evidence from a clinical trial that I was part of during my PhD, and talk about new MRI techniques that we can use to help us understand age-related cognitive decline and disease related to aging. So before we jumped into that, I'd like to start by just briefly mentioning a little bit of the epidemiology. I'm hoping that my slides move here. So I think everyone has seen this picture at this point, but it's important to emphasize that the number of people with dementia is rising. And this is growing very rapidly. This is from the Alzheimer's Association, and this is the projected number of people living with dementia in the US. And we see that there has been an increment, and the projected number is continuing to increase and increase over time. And by this time, which we are in 2025, it's estimated that more than 7 million people aged older than 65 will have Alzheimer's dementia. And this is a tremendous increase compared to just the year 2020, 5 years ago. So essentially, the whole point of this graph is to show how dementia rates are expected to rise and how a lot of the focus in ADRD or Alzheimer's disease and related dementias is into developing strategies to decrease the risk of cognitive decline and protect the brain health of our aging population. To build on the topic of Alzheimer's a little bit, I will briefly mention that Alzheimer's disease is the most common cause of dementia. And dementia is an umbrella term that we use to refer to memory loss and other cognitive abilities to a point that it can interfere with daily life. Alzheimer's disease is also the most common cause of dementia, and the most important risk factor is getting older, essentially. Increasing age is the most important risk factor for Alzheimer's disease. Here we see two pictures, and these are MRI pictures showing more or less what a healthy control would look like compared to an Alzheimer's disease patient. And again, this is a generalization because there are a lot of individual differences in how people present and how their brain structure looks like. But overall, in a healthy older adult, we do see some cortical atrophy, some decrease in the cortex of the brain, a little bit of enlargement of the ventricles, some changes in the white matter as well, which are not very visible in this MRI. But in Alzheimer's disease, we see a lot of these changes accentuated. We see a lot of atrophy that is evident here in the ventricles, a lot of cortical thinning evident as cortical atrophy, and many changes in the white matter that, again, are not visible in this type of image yet. And it is important for us to just have an idea of what are the expected changes in the most common type of MRI. What are the expected changes in Alzheimer's disease? But it's also important for us to know that Alzheimer's disease is considered to be part of a continuum. That is not that one day we start to have memory problems and immediately thereafter we get dementia, but instead it is a process that gets worse over time And now we know that even before we have any symptoms, there are already changes occurring in our brain. So even many years before we start developing any cognitive symptoms, we have some changes occurring in our brain. And that is a very important stage. We call it preclinical or before symptoms in this context. And there is a point in the disease in which some of the cognitive abilities start to become more impaired until we reach the point in which we're no longer able to conduct some of our normal day-to-day activities. And then we are within the spectrum of dementia. Although we do expect some cognitive changes as we age, these are not nearly as accentuated as we see in the context of Alzheimer's disease and related dementias. But of course, there's still a lot for us to learn in this context, but just trying to show you here more or less the comparison between what is expected in healthy cognitive aging versus Alzheimer's disease. So to talk about white matter, I'm going to start by just briefly mentioning what white matter is, because it is very important in the context of Alzheimer's. And I'll explain why, but first I'll just mention that white matter, if you see in this picture, we see the white matter versus the gray matter. The white matter, you can imagine as just being inside the brain, forming the connections that link different brain regions, and essentially helping ensure that we have a smooth cognitive functioning. And you can see in the other picture here on the right that if we zoom in into some of that cortex, we see these neurons that conform the cortex and some of these axons, myelinated axons that conform the white matter. And essentially those myelinated axons help make sure that we have quick and efficient signal transmission across different brain regions. So the important thing to note about this picture also is that even in the absence of cognitive symptoms, and even in the normal aging, we still see in the normal aging trajectory, we still see some changes in the white matter. Like if we take a microscopic picture of some of these axons and see some of the normal changes that occur with aging, we see a lot of damaged axons and a lot of axons that have lost some of that myelin. And overall, a lot of these processes contribute to some of the cognitive decline that we observe with aging. And some of these changes are also even more evident and pronounced in the context of Alzheimer's disease and related dementias. An extension of that would be this picture. Here we see normal versus Alzheimer's disease. And again, this is an oversimplification of a lot of complex processes. But in the left, we see kind of like a normal, healthy brain. We see the neurons here illustrated as well-structured, having intact connections. This is representing kind of the normal neuronal health and function. We see some of these cells called oligodendrocytes, and these are the cells that maintain the myelin health, maintain it and make sure that we develop new myelin and have that process of remyelinization. That is very important also to make sure that the signals are efficient. We have other cells called astrocytes that are kind of like a star-like shape, that these are very crucial for helping the neurons maintain and manage some of the neurotransmitters that help the signals at the synapse and maintain something that we know as the blood-brain barrier as well. Now, in Alzheimer's disease, on the other side, we see that a lot of these neurons, first, they don't look good. And we see a lot of these called neurofibrillary tangles. And essentially, these are aggregates of a protein that disrupts a lot of the neuronal function and contribute to the cell death eventually. But first, we see a lot of these amyloid plaques or clumps outside of the neurons. And these are like sticky accumulations that interfere with the neuronal communication at synapses and promote a lot of the inflammation that occurs in Alzheimer's disease. So essentially in this picture, we're just trying to show how there are some changes in glial cells. And some of these changes are very important and related to white matter. And we see that there are differences between what is expected in normal aging versus Alzheimer's, and that some of these inflammatory changes are in response to disease progression and actually worsen neuronal function and ultimately lead to cognitive impairment. But how does that relate to myelin, or the myelin in the white matter? So this picture is a little complicated, and the original article is here for anyone that wishes to read the original article. But essentially, in Alzheimer's disease, we have a lot of this amyloid beta that accumulates and forms plaques, and is mostly in the cortex of the brain, that gray cortical region that we observed earlier. And this causes a lot of toxicity, particularly in oligodendrocytes, which are these cells that are critical to maintain the myelin sheath, make sure that it forms. And the thing is that there is some direct damage that the amyloid beta does that impairs the ability of these cells to function. But also, as these cells get damaged and as the oligodendrocytes are kind of damaged and ill, they also cause a lot of processes, a lot of inflammatory processes, and release iron that also ultimately exacerbates and produces more amyloid beta. And overall, this kind of cyclical process leads to a lot of white matter lesions that we observe in MRI. But in MRI, sometimes we observe a lot of these lesions, but we don't know exactly what type of process is causing that specific lesion. But in MRI, we simply observe that something happened to the white matter, something is wrong with the white matter. The white matter looks pathological. It does not look normal. And some of these changes are related to losing some of that myelin. And as we lose the myelin, sometimes that leads to changes in the axons, and eventually we lose those axons. Ultimately, this leads to accumulation of bad proteins that lead to neuronal dysfunction. So now that we know that white matter is important to study white matter in the context of Alzheimer's, how can we study white matter is the big question, because we know that the brain is giant. We have 100 billion nerve cells and neurons, and all of them have axons. And essentially, how do we study something so big and so complex? One of the ways that we study the brain is using pictures, and MRIs are essentially pictures. And essentially, the MRI machine takes individual pictures of different parts of the brain at different cuts and kind of creates an image at the end using some MRI technology, and kind of reconstructs the whole structure of the brain. And sometimes these images are of higher versus lower resolution, and that depends on how small our voxels are. And the voxel is the pixel in this picture. So we use a lot of MRI to study the brain health and to also study the white matter. But essentially, we have two types of MRI that we use to study the brain: the structural MRI and the functional MRI. The structural MRI is the one that we will focus the most today, and it is the one that we use to study the white matter. Functional MRI is also very important, but it's mostly used to study how the brain activates in response to a stimulus. Like, for example, when we put a person in the scanner and ask them to solve like a cognitive task, like a puzzle, we can see how the brain kind of responds to that. And that is considered to be functional. It serves a different purpose, and it has different properties. But the structural MRI allows us to kind of see that cross-sectional picture of the brain and characterize these specific aspects of the structure of the brain. The most common types of structural MRI are, you can see them in this picture. These are the most common MRI sequences or MRI types that we use to study the white matter. We see the T1, the T2, and the flare, and the flare is like a type of T2. These are the most common types that we use both in the research world and in the clinical world to study the brain and to study the white matter. And we see, for example, that in the T1s, the white matter appears white, and the gray matter, the cortex, appears gray. As we expect, this is a relatively healthy brain. We don't see a lot of atrophy in the ventricles. We don't see a lot of cortical atrophy. In the T2 images, essentially the colors are inverted. The white matter no longer looks white, it kind of looks dark. And the structures that are in the ventricles, like the CSF or the cerebral spinal fluid, looks brighter. And essentially, we just use these different types of techniques to look at the structures in different ways. because if we have something abnormal in the brain, we want to be able to observe it in different ways and to kind of see what are the tissue characteristics. So that's why we use different types of sequences. The FLAIR image, I want you guys to remember that the FLAIR image is very important and has been very important to study aging. And the reason for that is a T2 sequence, but we suppress the fluid that it comes from the ventricles, and it allows us to see the structures around the ventricles a little bit more. And the structures around them are very susceptible to changes in the white matter. And that is essentially because a lot of vascular changes or small blood vessels change within those regions. So the FLAIR is a very common MRI sequence used to study the white matter. But essentially from any of these type of MRI sequences, we can extract different things to study. Things that we can derive, for example, in this image, we can see the white matter kind of highlighted in red and the gray matter highlighted in green. We can extract how much white matter volumes we have, how much gray matter volumes we have. We can also extract by regions. We can see the volumes of the different structures. We can see the volume of the frontal lobe, of the different parts of the frontal lobe. And essentially, this type of images allow us to see volumes, a lot of the volumes. Other type of images are shown here. This is a very important type of image and definitely changed the field of white matter and is something called DTI or diffusion tensor imaging. And this is a type of image that has different properties than the volumetric structural images that I just showed you, because what this tries to do is look at the water diffusion within the white matter. And the reason for that is that, for example, you can see in this image that the white matter is pretty evident. And that is because the white matter having so many myelinated axons, the myelin is composed of fat. So essentially, as we have a lot of myelin, the diffusion of water within that axon is going to go in a very specific direction because of that restriction. So this technique, diffusion imaging, has been a tremendously useful technique for us to advance the field of white matter and understand how white matter changes as we age. And I'll explain a little bit more about it in just a second. But essentially from these diffusion type of images, we can extract more sophisticated techniques, such as this one here called tractography, where we essentially see where all the tracks of the white matter are going. And this is a type of modeling. And it's not, sometimes it's very precise, sometimes it's not, because it depends a lot on the techniques and the models and the type of analysis that you use. But we so far have discussed the structural MRI techniques, the most common that we use, the T1, the T2s, the FLAIR, and now DTI being the gold standard technique that we use in research. So talking about DTI a little bit, this is that DTI picture that I showed before. And as I mentioned, to date, this DTI technique has been the most important technique used to advance the field of white matter in the context of cognitive aging and Alzheimer's disease. And it measures the movement of water molecules within the white matter. And the most common type of measure that we use from this is something called FA, or fractional anisotropy. And what it does is that it kind of calculates the main direction of water movement within a white matter voxel. Here, like in this example, like in this voxel. And depending on how many myelinated axons and how many things are going on within that axon, we can see the white matter or the water kind of moving in one direction. So for example, in aging, because there's a lot more demyelination and axonal disorganization and other inflammatory processes, we see lower FA values, meaning that the water moves more randomly. And that also happens in other processes, like in the processes that we observe with Alzheimer's disease. More demyelination, so water moves more randomly.
Andrea?
Yes?
Yeah, I'm sorry. Could you go back
Yes.
So is this image, it looks kind of pixelated.
Yes.
And how is, I mean, why we see the image like that, and when we are looking for more detail, uh, anatomical changes, and the practical question is, so is this really how much like the type of magnet, like 1.5, 3, or 7, it make a difference in this type of technique?
Yes, those are all very interesting questions because the type of magnet does help with the increase in the resolution, and it allows you to have smaller voxels that will allow you to have better image resolution. So definitely 3 Tesla is very good. Maybe 1.5 is not as good. 7T would be very, very good, more specific, a lot better resolution. But all in all, what matters the most is the way that you analyze these images to derive this FA number, and making sure that your data is well pre-processed and that you are using a pipeline that is well studied and standardized in a way. That is what really makes a difference in calculating some of these numbers, when you compare, more than comparing the different types of MRI scanner quality. But in terms of why the image looks pixelated, the reason is that this image is moving in real time, but I just took one picture at one time, because when you acquire these images, the diffusion, you can see it happening in real time. And you kind of extract these numbers from each of those voxels and from the white matter tracks, but you don't analyze these images qualitatively, but instead you analyze them quantitatively. Like, you extract numbers from them, but you don't analyze them just to the naked eye, because that's more of like what we do with the T1s and the T2s in the clinical setting. But in the research setting, we instead extract numbers from these voxels, which we're not able to see in this image right now, because if you're acquiring the image in real time, all of these slices would be moving.
So when you say moving, is it like the atoms that are spinning, or it's at a more like water type of level?
More at the water type of level, we see some of the diffusion going in multiple directions, and we just see the contrast because it's in the image, in the scanner. When you're acquiring these images, you just see black and white and grays contrasts. You don't see the sophisticated tractography images with all the colors. That would be like a post-processing thing that you do after you acquire all your numbers. But in real time, you just observe kind of those water molecules moving in within the voxels, and it's all very noisy because you're just seeing all the water kind of moving through the different aspects of the white matter.
Thank you.
No problem. All right, thank you, Dr. Gladys. Those were really good questions. So I think that's a very good point about DTI, and everything that Dr. Gladys mentioned is really interesting. And again, DTI, this is the most common technique used in the past years, but it doesn't mean that it's perfect. And the truth is that there is no perfect MRI technique. And the reason why it is not perfect is that we can see the water diffusion going along the axons, but sometimes there are so many microstructural changes happening in the tissue that we don't really know what is causing the water to move in one direction versus the other sometimes, because I showed you some of the processes that happen with Alzheimer's, for example, but the reality is that in real time, in vivo, there are a lot of pathological processes that happen and that can change this water signal. So it's a very good technique. Again, has allowed us to make a lot of changes to the field, but it's not perfect. One good thing about DTI, however, is that it's very good at detecting how the white matter changes with age. is very sensitive to age-related changes. And we already know that white matter changes as we age, but the DTI-related techniques, particularly that FA or fractional anisotropy, has a very strong relationship with age. And because of that, it correlates very well with cognition. But sometimes that correlation with cognition is kind of confounded by the effect of age. So that is a very important thing. And it is why sometimes the DTI studies have shown some consistent and some inconsistent results. Another way that we study the white matter that is very common is using those T2 images, particularly the FLAIR images, because we can see some of these lesions here. And you can see some example of like early stages of the lesions and some like more severe changes of the white matter surrounding those ventricles. And from those images, those FLAIR images, sometimes we can derive something known as white matter hyperintensities volume or white matter hyperintensity volumes. And we can extract how and derive how many of those lesions we have in the brain. But again, we are taking a picture of those lesions and we are seeing how many white matter hyperintensities have, but they all look very different. Sometimes they're related to one process, sometimes they're not, sometimes they're related to demyelination, sometimes they're related to more changes in the vasculature, and they're very strongly correlated with small vessel disease. And sometimes these changes can be reversible and sometimes they're not reversible. So this mechanistically, this technique, white matter hyperintensities extraction, is very good. But again, it represents a lot of different pathological processes that happen in the white matter as we age, and a lot of pathological processes that happen in the white matter in Alzheimer's disease and related dementias. But the big question, can we slow down white matter decline with lifestyle changes? And I know I've spent a lot of time talking about MRI sequences, but I guess it is because it is very important for us to know what we used to study the white matter. But most of what we know from the white matter and from the pathology side of things, it's coming from animal studies, mostly rodents. And from animal studies, we know that a lot of experiences that are positive, like motor learning, social interaction, physical activity, all of those like lifestyle or experiences can cause changes in the cells that create the myelin, and eventually this experience derived or experience dependent process that happens throughout people's lifespan, or in this context, this mice lifespan, can lead to more myelin eventually. So we see different myelination patterns depending on lifestyle. But a lot of this has been tested in the context of animals, but not yet as much in the context of humans. And the thing is that in the context of humans, we have used a lot of the more traditional techniques, but we have not yet studied the white matter decline and how the white matter changes using more advanced MRI techniques. So yes, white matter changes. We can prevent some of that white matter decline that happens with lifestyle, but it depends and depends on a lot of things. There's a lot of heterogeneity in the results that we have in the literature, and I'm going to show you one example. I'll skip this and show you instead this example using white matter hyperintensities. This is a study in which they followed people over time, baseline 1 year, 3 years, 5 years, and we see that this is qualitative. We see the images, we see how many white matter hyperintensities there are and how they continue to increase with time. Sometimes we see comparable changes between subjects, but sometimes we see that these white matter hyperintensities don't develop in certain subjects. So the truth is that a lot of the studies have shown that white matter does improve with exercise, but there's a lot of variability in the results. And a lot of the variability is also due to things such as what we see here, that it depends on ultimately how much exercise you do and of what type. For example, this is like moderate to intense physical activity, which is what the American Heart Association recommends. And we see that there's kind of like a point in which we start to see that the white matter hyperintensities stop growing and the white matter essentially becomes healthier. And we also see similar things with sedentary time. So this is just to kind of give you an idea of why we have had so many null results and so many inconclusive findings related to white matter and lifestyle, specifically physical activity. So now I'm going to transition and show you one of the studies that I published during my PhD. And this is a study using an MRI technique that is an indirect measure of myelin, and it is used calculating the contrast differences between the T1 and the T2 images. And essentially, this is an indirect measure. It's not the most updated or new measure to study myelin. I have some examples of those in next slides, but it's just to show you that maybe targeting different aspects of the white matter will allow us to see how white matter changes with exercise. And essentially, the conclusion of this study was that the white matter regions that are vulnerable to aging have some plasticity, meaning that they change in response to exercise. This is the image type that we used, the T1-T2 ratio, and is essentially taking advantage of the difference in contrast between the T1 and the T2 images and kind of isolating the contrast related to myelin. And we can see some of this myelin highlighted in this picture. So in this study, we had 180 adults aged between 60 to 79 years. And these were healthy, older adults, cognitively healthy, without significant risk factors, with controlled controlled hypertension and other characteristics similar to that. And this was a clinical trial in which we had 3 exercise sessions lasting 1 hour per session per week for 24 weeks. So more or less 6 months. And we had 3 groups. We had the active control, and which active control means that people still came to the institution and were still involved in some level of activity, but it was not exercise per se, but more of like stretching exercises. We had a dance group and we had a walking group. And all of these images, and all of these participants had MRIs collected before and after the intervention. So essentially, the aim of this study was to report the effects of a 6-month aerobic walking and dance intervention on the white matter integrity in healthy older adults measured with the T1-T2 ratio. Some of the things that we expected to see is that in people that were exercising, meaning people within the dancing and the walking group, we were expecting that the white matter was going to get better over time. And we expected that in those that were in the control group, the white matter was going to get worse over time, just as kind of representing the normal processes that we would expect with the white matter. These are some of the results and the outcomes of this study. And I'm going to walk you through some of these as an example. In the total white matter, we observed that both in the dance and in the walking, there was an increase in white matter, let's call it like that, an increase in white matter health over time. And in the control group, there was a decline. In some areas, we observed that there was always a decline, but in those that were exercising, the decline was kind of diminished compared to those that were not exercising. And we observed this effect particularly in regions that are vulnerable to changes with age, such as the most anterior part of the corpus callosum, which connects the frontal lobes. We saw that there was an increase with exercise in the white matter. And now, did this increase in exercise relate to better memory? Actually, it did. But only in the walking group, not in the dance group. Maybe because in the walking group, there was more time spent exercising. The dance group spent a lot of time with instructions. Maybe the exercise was not of the same intensity as in the walking group. But essentially, we observed some increase in memory over time compared to the control. And this is all important, but again, this was a 6-month study, and we were looking at cognitively healthy participants. It was not a very diverse group, and they were, again, very healthy in one specific region of the US. So, of course, this was a very promising finding that we had, but there are a lot of things that could add to different results in other populations. More recent evidence, however, and this is from a group that is at the NIH, they observed using a more sensitive MRI technique to study myelin that in the groups that were exercising the most, and this is a measure of how intense your exercise is, they had greater regional myelin, especially at the middle ages and at the older ages. So we can see here 60 to 79, 80 to 94, and we can see some changes in the myelin content as people are exercising more compared to when people are exercising less. So this is very recent. It was just published a few months ago. And again, it's a little bit more evidence that there is some myelin plasticity in response to exercise. But another question would be, can some of these new MRI techniques help us understand the association between white matter and cognition? And the reason why I bring this up is that sometimes we see MRI changes in an intervention, but we don't see changes with cognition. So this is clearly very important for us to know how much of the changes that we observe in the white matter lead to better cognitive function over time. And this is results from another study that I published during my PhD and my postdoc, but this is using cross-sectional data. It's not using, it's not a clinical trial, it's not a longitudinal study. But in this study, the findings were that myelin measured using a myelin water image, newer MRI technique, but not the traditional gold standard FA from DTI, mediated the association between age and memory in healthy older adults. So I think we remember that I mentioned that DTI is the gold standard image that we use to study the white matter. And the reason why we use it so much is that it correlates with aging very strongly. And as we get older, the fractional anisotropy gets lower, because we expect more things like demyelination to be occurring as people age. And we see kind of similar patterns with cognitive functions as well, that there is more like a curvilinear relationship with age. But there are still a lot of gaps. And as I mentioned, no technique is perfect. But in the context of DTI, a lot of the knowledge that we know is from histological studies and animal models with small sample sizes, looking at very limited brain regions. And the in vivo studies that we are performing in humans rely a lot on the DTI technique. And because, again, there are a lot of processes that can be happening within the white matter, sometimes we find inconsistent associations with DTI and cognition. So ultimately, there's poor evidence for myelin loss in vivo and how it associates with cognitive decline in humans. So there are new techniques emerging, like this one over here called myelin water imaging. And we see the image here. And again, this is an image that we don't study necessarily qualitatively, but instead we derive some metrics from it. And in this context, this myelin water imaging works because the myelin, as we see in this cross-sectional picture of axons, is organized into these like concentric layers around the axons. And between these layers, there are spaces where water molecules can reside. That is what we know as myelin water. But there's also water within the axons, so there's also water within the extracellular compartments. So essentially, this type of MRI calculated the myelin water fraction by isolating the signal from the myelin water and expressing it as a percentage of the total water signal within a voxel in a brain tissue. So I know that's a little complicated, but essentially it allows us to isolate the signal related to myelin a little bit better than traditional techniques. So in this study, we used that myelin water imaging or myelin water fraction to investigate the role of myelin in mediating age-related cognitive decline across the lifespan. And our main hypothesis was that myelin in the prefrontal cortex, which is one region of the brain that is very susceptible to aging, and the gene of the corpus callosum, but here I'm going to focus on the prefrontal cortex. Myelin in the prefrontal cortex mediates the effects of age on cognition, meaning that we're investigating essentially how myelin, specifically in the prefrontal regions, influences memory as we age. So when we talk about mediation here, we're looking at myelin as kind of an intermediary. So we want to know how much of the impact that aging has on memory can be explained by changes in myelin content. So first, before we answer that question, as a sanity check, I just plotted to see how age relates to cognition. I expect cognitive functions to decline as we age, and that's essentially what we observed for memory, processing speed, and executive functions. But if you see, it's not exactly a linear process, but it's more like a curvilinear process in which sometimes there can be some peaks, but this is depending on the population, but sometimes it's a little bit more linear. But overall, we expect cognitive function to decline with age. But now, how does myelin relate to memory? And from this graphic here, I want you just to see those things that are highlighted. And essentially, we observed that the myelin in the prefrontal regions of the brain was very good at mediating the effect of age. And this is a coefficient that can be interpreted from 0 to 1. Closer to 1 is stronger in effect size. So myelin was very good at mediating the effect of age on memory. But FA, which is the gold standard derived from DTI, was actually not when we included in the same model and adjusted for the effect of age. It looks like age is so strongly correlated to this traditional MRI technique that we lose the effect of it in relationship to memory. So essentially, in this study, we observed that more myelin in the prefrontal white matter relates to better memory. And here is kind of a replication of those results, but using a different type of analysis. And we essentially, those regions that are kind of highlighted in yellow were the regions where we observed the strongest effect with memory. So myelin in those more like prefrontal and frontal white matter regions. So essentially, can advanced MRI techniques help us understand disease mechanisms? I will say yes. But again, some of these techniques are still new and in development. And I just showed you an example of our study looking at myelin imaging. But there are more advanced techniques coming up and that are being used. For example, within the ADNI project, they're using more advanced diffusion MRIs. And this is an example of that. There is, oh, I skipped a letter here. There is a new diffusion MRI technique called NODI that is the evolution of DTI, because I think we discussed earlier how DTI, as in the far left in this picture, kind of looks at how much the water moves within an axon. And it just tells you what is the main direction of the diffusion of water within that axon. But more advanced MRI techniques are looking at the water diffusing in different places within the axon, and that kind of can help us understand some of these inflammatory or pathological processes that can occur with aging and with ADRD. This is a new technique, and I will give you one example from our publications. This is a publication in which we wanted to see how NODI and myelin associated with metabolic syndrome. And I'm not going to show you the results per se, but just kind of like a summary, that when we used myelin water imaging, we observed that more myelin was associated with a decreased risk of metabolic syndrome. But when we use NODI, which is that advanced DTI, kind of advanced DTI technique, we observed that it was not associated with metabolic syndrome. And again, because this is cross-sectional data, we don't really know what's the causality here, but our conclusion was kind of that metabolic syndrome maybe first exerts negative effects on the myelin, and that later can be associated with deterioration in axons at later stages of the disease. So to start concluding, I want to kind of show you some of what I expect to be the future of white matter MRI. And this is some of the things that I expect that will happen in the future or are happening right now, is that I expect that we will combine some of these quantitative metrics for a more specific assessment of the mechanisms of white matter decline and associations with cognition. I expect that this will help us understand better the interplay between small vessel disease, demyelination, and cognitive decline. I expect that linking MRI findings with some of the fluid biomarkers that we use to define Alzheimer's disease will help us identify some different myelination processes. Also, using longitudinal designs will help us understand how early white matter changes that happen earlier in that continuum of aging and continuum of dementia, how those early white matter changes relate to cognitive impairment. And obviously, we want to see how these techniques in the future can help us guide intervention, hoping to see remyelination therapies and vascular risk reduction, and seeing how MRIs can help us assess the treatment response in clinical trials. And a very important point, lastly, is that I expect that in the future, we use advanced white matter imaging to study diverse cohorts to reveal how genetics, lifestyle, and social determinants of health and comorbidities affect white matter changes. So to sum up, today we explored how white matter declines linked to Alzheimer's and related dementias, and we emphasized the role of myelin. We discussed the use of advanced MRI techniques and traditional MRI techniques like T1-T2, T1-T2 ratio, white matter hyperintensities, the gold standard diffusion imaging, myelin water imaging, and more advanced diffusion techniques. We highlighted how the aerobic exercise, can influence white matter integrity, demonstrating that the brain, the white matter, can change in older age. And we discussed some future directions in white matter MRI research. So before I finish and show you my thank you slide, I want to say that here at Duke, I'm currently working, studying disparities in aging research, and we're looking for a postdoc. So I want to show this publicity here because I would love to see some of you applying for this position. Dr. Maria Marquin, who is my advisor for my physician scientist track at Duke, is absolutely fantastic. And we have this group forming here at Duke, and is totally recommended. So I'll just stop there and say thank you all and open for questions.
Thank you, Dr. Colmenares, for the wonderful talk. Very detailed, very interesting. For the MWF, what sequences are you using in your center?
Mm-hmm. That is good. Let me see if I have, actually I have another slide where I can show you a little bit more in detail what sequences I'm using. This will just take me a few seconds.
Andrea, and so Anderson is not only a good friend, also coming from Brazil, but he is an expert in imaging, and so came less than 2 years ago, right, Anderson?
2 years ago, yeah. But
2 years ago.
today the expert here is Dr. Colmenares. I'm here to learn only, right?
So he handles the magnet too. He has a nice magnet. Which one is the one you have, Anderson?
It's a Siemens Vida 3 Tesla.
Oh, fantastic.
Yeah,
That's amazing.
very
And what
easy.
techniques are you using, Anderson?
So the scanner was installed it was installed about a year ago, but we only started using it in August. And we don't have any studies running. Actually, Gladys, you are going to be the number one to use our machine. So there is a good deal that we need to do. For the MWF, I asked because in my previous place at the NIH, we did collect using MacDespot, and we
Yeah.
processed those images using Quit. There are other tools available, but we found that Quit was easier and more direct to use. But there are issues with optimizing and finding the correct parameters and computing the correct parameters. Are convergence failures and a number of issues, and even with Quit, right? So that's why I ask if you have an alternative, if you're using anything different than MacDespot, that we could also give it a try.
Actually, I think you may want to reach out to Yongho Lee in Korea because he has this VISTA myelin water imaging technique that we initially tried to apply, but we didn't have an MRI physicist on site in Colorado to help us set it up. And we acquired some images actually using this new technique, and it's a fraction of a short T2 star signal over total water signal, and is faster than Despot, very easy to analyze, is a little bit more sensitive, you see, compared to Despot, just even qualitatively, just to look at that myelin water fraction. But again, the reason why we couldn't use it in Colorado is because there was a lot of artifact, probably related to the pulsatility of the carotids coming up from the brain. So we saw a lot of vascular artifacts in this image. So we couldn't really use it, and we couldn't really update the parameters in our scanners. So we ended up using the same parameters that you're using right now with Despot.
Okay.
So it's still T2 based, but Vista, very worth looking into, and Yongho Lee may help you with all the setting up and making sure that you can apply it and implement it.
So can I send you after this conversation, after this meeting, can I send you an email asking his contact details?
Yeah, absolutely.
All right. Thank you. I'll do that then.
Thank
Thanks.
you.
So, Andrea, now that you mentioned the pulsatility, what really does that mean? Means like individuals with higher pulse pressure will have more artifacts?
It's hard to say because this is just what we were observing, that in the point in which the internal carotid kind of bifurcates, as well as in the vertebral artery, and in real time, when there was any sort of pulsatile or any sort of movement in those regions, we would get distortions of the image. So I don't think it's necessarily that we were looking at any direct associations between pulse pressure or increased pulsatility and what myelin in real time, but it was more of like an actual artifact of just trying to isolate some of the image signals. And sometimes it's really hard to do because the brain has a lot of noise and these
No, what
images are hard.
I ask because some of our colleagues do postmortem brain imaging, and white matter hyperintensity, of course, is one of the neuropathological aspects they look, and they have the personal life because they've been part of a longitudinal study, and then they get the postmortem brain and put it in the magnet. And when I ask how are the associations, there are, of course, a lot of things that the agony and the time between the MRI alive, because obviously the patient was not so deteriorated, but most of them agree that the agreement is bad.
And what do you mean by the agreement, Dr. Gladys?
That what they see in the white matter hyperintensities and the level it's so they don't find what they expect in the postmortem.
Hmm.
And I say sometimes it's worse. I assume it's worse. I say sometimes, sometimes not. So
Mhm.
I'm just thinking, what is different between, of course, many things are different between a dead and an alive brain, but just thinking how the pulsatility of, I mean, the circulation of the blood might be able to, and the differences between this pulsatility between people will actually affect the quality of the acquisition.
That is a really good point. And I actually don't know if there are these type of artifacts observed in the FLAIR images that you use to calculate white matter hyperintensities. But I certainly know that the algorithms to calculate white matter hyperintensities have been developing a lot more recently. And there are more like automated and semi-automated methods to extract those images, because the initial way in which white matter hyperintensities are extracted is that essentially people draw the white matter hyperintensities by hand, and then they generate a mask out of those and essentially just extract the volume of those white matter hyperintensities in each brain. And that is, of course, there's a lot of interrated variability sometimes, as well as a lot of space for making calculation errors. But I know that more recently the techniques have changed and there are a lot of new ways to calculate those. So I don't know if that is sometimes introducing some of that variability versus more of like artifacts during the acquisition. I think it's more related to the method itself and how it was calculated.
But when you show the improvement of the white matter in exercise, did you adjust for blood pressure?
Yes, I did, but this was a healthy sample with not a lot of variability in blood pressure, unfortunately. So I couldn't really see an effect, and it didn't really adjusting for blood pressure didn't really change the results. So I will always wonder, and I wonder if this has been done and will be done, how it would look like in a population that has hypertension and different patterns of blood pressure.
Yeah, the different patterns, I think, and also the blood pressure response to exercise,
Mm-hmm.
because it would really kind of influence the vessels in the brain.
Mm-hmm.
So thank
I
you, Andrea.
agree.
I think that was very, very interesting. I see Dr. Martinez.
Hello. Great presentation. Unfortunately, I came in a bit late during the presentation because I had some other businesses to attend to. But I did wanted to ask about, I think I came in right as you were talking about the slide where you were comparing the walking intervention and the dancing intervention. And one of the things that I guess you mentioned that the dancing intervention didn't exactly have the same there wasn't a significant effect on the degree of white matter degradation with aging comparatively, and you were thinking that that was maybe because there was less time actually dancing as opposed to just, I guess, standing or sitting while receiving instructions is my understand. Do you happen if in terms of the details of how the study was done, do you recall like how many minutes was people like required to be walking? How many minutes were people required to be dancing? Also, if the like let's say in the dancing intervention, was this kind of like dancing or music that people were already familiar with, or that they were unfamiliar with? And the reason why I'm kind of asking into these kind of seemingly nitpicky details is that kind of in relation to the as you were linking to the loss of what is it, white matter integrity in the prefrontal cortex. A lot of that is essentially related to executive functioning. And if depending on, especially with like the dancing interventions, let's say, if it's something that is completely new and foreign to you and not something that you've been doing, let's say, um, throughout your life, um, it is something that's going to require a lot more use of the the prefrontal cortex, and basically more frontal white matter activity. So I'm wondering if this would also maybe there is some, just kind of picking into more questions, there's maybe in terms of the quality of the comparison of the two interventions, if that maybe also further alters how you would see things, or how things would come out.
Thank you. I think there are a lot of things to disentangle from that, because the thing is, one of the few things that immediately came to my mind is that yes, some of the type of dances were new to people and they spent some more time learning, and that was still considered to be part of the 60 minutes that was allotted for the intervention. But the thing that was measured very accurately was with accelerometer data, seeing the intensity of the exercise and the VO2 max, which is kind of like a proxy for knowing how intense the oxygen consumption was during exercise, and things like that. We looked at, and we didn't see those being as high in the dancing group compared to the walking, which is why I'm kind of like attributing it to the intensity of the exercise, which is something that we also observed in the example that I showed with white matter hyperintensities, that maybe there's a point in which it depends on the type of exercise, on the intensity of the exercise, along with maybe even environmental contributions, because the people that were walking were also walking outside. Maybe they were engaging in social conversations. Maybe they were seeing the trees. Maybe they were like involved with the environment a little bit more. And that aspect of environmental enrichment added, I guess we didn't really account into, and we didn't really measure it for us to know whether that had any influence. But there are many maybes, and the truth is that it could also just be in a sample size effect, because the dance group was just underpowered compared to the walking group. So we did observe the dancing group having a positive effect in the white matter. We just didn't see that relate to better memory like we did with the walking group. And again, I don't think that needs to be a strong conclusion. I think it just shows that there are some things that maybe we didn't measure, or more talking about the design of the study.
Okay. No, that makes complete sense.
Thank you. Thank you for your question.
I like the suggestion of the type of music. Because like the samba, like Anderson will attest to, the dancing might be different. I mean, for how long will you be able to stand dancing samba? But I also make me think that when you dance like with somebody, like it's in a salsa type of, it's different that when you dance alone. While walking, it's kind of the same in terms of, so did your participants dance alone? I mean, like in front of a mirror, or this was couple?
And by the way, I
Mo-
know Dr. Martinez is a great dancer. So
Awesome.
Thank you, Dr. Maestres.
Well, it was a combination of both, like more of like a bailoterapia type of setting, as well as like dancing together. But we also didn't like discretize that, and I don't think the sample was big enough for us to see like differences within the dancing groups. So definitely worth looking into, because I think the social aspect definitely makes a difference there, but I don't think that we were able to answer that particular question, and I don't know if there are yet studies looking into that specific aspect of the physical activity and the how it relates to social interaction as well.
and the other thing that Dr. Martinez mentioned that I thought was very interesting was the association of the executive functioning, perhaps because of the prefrontal cortex and what you were saying, to the, I don't know, difficulties in dancing, or that will be sensory motor um, involvement as well. I mean, there is something about kind of, I don't know, motor memory. Um,
Yes, so it would be, so what I was thinking is if you have, depending on how one's level of competency and proficiency, if somebody is a novice to you would be using a lot more executive functioning versus um if you're doing salsa sessions and you had been doing salsa your whole life, like if my mother was a participant in one of these training sessions, it would be completely from muscle memory, as opposed to if I were to start taking swing dancing classes, which is something that is completely foreign to me, it would take a lot more of that frontal executive functioning. And so that's why I'm kind of using those differences just in terms of you are essentially exercising different aspects of your cognition. And that's why I was wondering if and since there was the link with particularly with the frontal white matter behind the prefrontal, associated with the prefrontal cortex, if maybe there was any, if maybe the novelty or the difficulty of the intervention had any sort of effect on just kind of the outcome. At the same time, it could also be a vice versa thing where because it's a more difficult thing, you're spending more time kind of just like learning the instructions and not actually doing the intervention. So there's kind of a pro and con to it. Um, and that's kind of what I was trying to tease at.
no, very interesting. But I have a question for you. Do you think, Dr. Martinez, that imaginary dancing will, have the same effect in terms of the connection? Like if you visualize that you are dancing, like paraplegic person will imagine, and you can imagine yourself dancing.
Good, very good question. Um, that's I don't know if it would necessarily it may have some effects on frontal. That that's actually something I'd have to definitely ponder about a bit more. It is a stump worthy kind of question that but you might be still exercising some of the, if you're imagining things with like a visual spatial component, you might still at least be exercising those parts that have like let's say your parietal lobes, which are involved in sort of more of that visual spatial memory, those will probably be activated.
So even if you are very lazy, will it be good just to perhaps watch a video or imagine or follow instructions about we have a colleague, we had her a couple of months ago here, showing the effect of Qigong or Tai Chi, imagine like just imaginary on pain. And basically she found the same elements. It didn't matter if the person could not actually or could not or would not do the physical exercise. It's just the brain in that case was the body awareness. That's how she put it together, the body awareness. And but needed to be somebody with high attention, so he can follow the instructions and be following, which will be a little bit harder with dancing. But I'm gonna try to daydream more about dancing, maybe. That's something.
I wish I had listened to this discussion before I wrote my discussion.
What do you think, Andrea?
Of the imaginary part?
Yeah.
I think we could definitely see it influencing structural connectivity and maybe functional connectivity as well, but probably would not relate to the same effects that we observe with physical exercise, but yes, would modulate some of those pathways that are involved in specific things such as pain. But again, I think it would be very, very complicated to find people who have that level of attention, because it's kind of like a mindfulness type of intervention.
Yes. And what about musicians? Because musicians kind of imagine, and to let you all know, Andrea is an amazing singer and play the guitar so beautifully. But yeah, and now that we have even Nay here, with more complex, formal classical training, music training. But it's been shown that playing, I mean, imaginary, going over it's basically the same as actually playing. I remember these functional studies in Bruce Springsteen and other classical musicians. It's an imaginary going over partiture for, at least for brain activation, was basically the same, but those only work in people that were experts, not in people that were just novices.
I imagine that at that point, when you're capable of improvisation with music, is kind of knowing like another language, and similarly to knowing another language, that also contributes to cognitive reserve and maybe some of those effects.
What do you think, Neil?
I think that there is an important role of the endorphins here too. So it's not only the exercise. The exercise is very important, but also how the mood is modified by doing exercising, by doing dancing, by playing with music, especially if you like that music. If you don't like, maybe something annoying, but definitely if you like the music, the reaction is potentiated by the endorphin system. So definitely hyperproduction of dopamine and other metabolites that are going to be raising in that brain. I don't know how much they can help to reconstruct some pathways or some myelin, but it's possible. There is a big hope. I like that idea, definitely, and count with me for anything that if you want to have another site to do analysis of this type. Definitely going to be interesting. Yeah, I'm actively involved in the music research too in the brain. So this is beautiful,
Fantastic.
really beautiful, beautiful. I have another question. Was about what program are you using to extract the data from there? I saw that Nodi has its own procedure. It's something relatively new from Europe. Are you using also a free surfer or other method to compare the white matter hyperintensities?
So I didn't do a lot of white matter hyperintensities work, but when I had white matter hyperintensities extracted, I did use free surfer.
Okay.
For diffusion related images, I used a lot of FSL.
Okay.
And for Nodi, I use a MATLAB toolbox called, I think, Nodi MATLAB toolbox. Exactly.
Yeah. No, that's not beautiful. Yeah, we have here. Well, I think the most widely used is free surfer. Yeah, that's important. Well, I think this has a lot of potential, and yeah, the development on this area is going to grow exponentially the few years. You're doing that. Good for you, Andrea. Very nice. Proud to see
Thank
you.
you.
Yeah.
Very, very nice. And I want to mention that Dr. Martinez is a behavioral neurologist, is our new neurologist. So take the time to get to know this group. And maybe when Andrea is done with her residency, we are going to have her here, hopefully, at least before you come with to visit and consider us as a potential place. So we start talking about it right now, and Dr. Martinez also will be the director of the residency that should begin, I think, 26 or this year.
Actually 25. So
Amazing.
we'll have our inaugural class this upcoming July.
Oh, that's amazing. Congratulations. That's fantastic.
Yes,
Thank you.
very, very exciting times. And I notice Jesus is here, the real Jesus, the real Jesus is here. But I know he was in an appointment
Hi. Hello,
meeting.
Andrea. I'm familiar with Andrea's work, and I would add to the curriculum of the university, and now I just wanted to make a comment about the imagination. So, with the Gladys was mentioning, so what about people who are writers? I mean, she was talking about imagination, and if you have to picture a story, I don't know, Harry Potter, or Lord of the Rings. I mean, you don't necessarily are manually stimulating your brain, but certainly your imagination is go beyond that. So it should have a positive impact. And before commenting that, I wanted to Nate also mention about the possible reaction if you listen to positive music. So I think if you measure now my brain activity with all the vallenato I listen to during Christmas, I think it's not gonna be positive. But yes. That's right.
Without dancing, that's the problem. If you listen to music and you don't dance, that's the problem.
Yeah, no. The writers, I mean, how their brain will work, and they are not doing something manual, but definitely imagination thing. It's there. Yeah.
Good. And the other thing, just to close, is so both physical capacity in terms of fitness for older adults, but also in terms of mental, like meditation, that kind of body awareness that you acquire during life through different things. So it will be very interesting to see how these things might help to produce positive effects in white matter.
I love that idea. And in fact, I have a friend who went did the PhD. He was part of my PhD class as well in Colorado, and he's now at MGV, actually looking at a similar question in the context of mindfulness and actually more into like functional MRI, but also a little bit of white matter. So I think there are people actually diving into these questions more recently now with the growth of mindfulness and meditation practices.
The effects are so powerful, and still we do not know how they work really and how to maximize, and they are usually cheap. So
It's true.
thank you very much, Andrea. It's been amazing to have you, although I have to commit, I have you to commit to play for us something next time in real time. It's so
I would
beautiful.
love to visit sometime. Thank you so much.
Thank you. Definitely when you come, it will be beautiful to have you and Nate to play together, and also Nate has a sister that is a professional flautist, and oh my gosh, she makes that flute really speaks and sing so beautifully. So another advantage of coming here, a lot of music, and the valley, it's really a very musical place. So whole Texas is in a way. And of course, Dr. Martinez is from Corpus, where our beloved Selena was born. So, it's a lot of music around. So thank you very much, Annie.
Thank you so much, Doctora.
Okay, we have the announcements for January. So we have an annual meeting of the RECMA. So the deadline to register is going to be in March 14, but we have, okay, it's here, the TARC consortium going to be next week. And many of us are going to be going there to see a good representation of UTRGV. We also have report release webinar on January 15, this is tomorrow, of the National Academies, research priorities for preventing and treating ADRD. And we will have our next presentation of this series of seminar on January the 28th at 3:30. The presenter is going to be Dr. Ryan Oprea. We're going to be presenting on decisions on the risk or decisions on the complexity. It's going to be a
Thank you, Nay. And I have to add that Dr. Prea is an expert in how suffering affects decisions.
Mm.
So I think it will be a very eye-opening, because his publications are just like very creative and very robust in terms of what we know about suffering, origin of suffering, and consequences of suffering in the brain. So thank you very much.
Thank you.
And see you soon. Thank you. Thank you, Neida, for
you.
everything you do for us.
Yeah,
Thank
and
you.
Thank you, everyone. It was great to see you.